In this mini lecture, we will cover the first phase of glucose oxidation, which is glycolysis. Firstly, let's begin by doing an overview of glycolysis, the metabolic pathway. Glycolysis occurs in every cell. The rate changes depending on the demand for ATP and glucose availability. So the energy status of the cell really drives how fast and how much we use glycolysis. If the energy status of the cell is low, we need energy, and therefore the rate through glycolysis will be very fast. If the energy is enough within the cell, then glycolysis will operate slower. Remember, the substrate that enters glycolysis is glucose, and the end product are 2-pyruvates. Glycolysis is the splitting of glucose into two products, which is 2-pyruvate. As you can see by the equation on the second point, this is the inputs and outputs, or the substrates and the products of glycolysis. We have glucose, C6H12O2, plus 2 ATP. We have 2 ATP coupling reactions, plus 2 NAD+, which leads to 2-pyruvate plus 4 ATP plus 2 NADH. This is a very important equation, and it summarizes what exactly the products are, the reactants and products of glycolysis. Glycolysis has two major functions. One is to generate energy in the form of ATP, and this is through substrate level phosphorylation. The second is to generate pyruvate for final oxidation in the mitochondria, along with NADH, which is the substrate used in the electron transport chain for oxidative phosphorylation. The overall Gibbs free energy of glycolysis is negative 19 kilojoules per mole, which means its overall reactions are exergonic and therefore favorable. There are three main types of reactions in glycolysis. The first one is hydration and dehydration reactions, which, as we defined previously, are the addition and or removal of water without splitting or cleaving a molecule. These are accomplished through a hydratase and a dehydratase enzymes. The second type of reaction in glycolysis is oxidation or reduction. This is also hydrogenation or dehydrogenation. So we lose or gain electrons, or hydrogens for that matter. So the enzymes are dehydrogenase enzymes for these reactions. So NAD plus and FAD are the coenzymes that are required for these reactions. And then the third type of reaction in glycolysis is phosphorylation and dephosphorylation, where we lose or gain an inorganic phosphate. And the enzymes that catalyze these reactions are kinase and phosphatase enzymes. Now let's look at the big picture overview of glycolysis. The net result of glycolysis is that one 6-carbon glucose molecule is split into two 3-carbon pyruvate molecules in a series of reactions that both consume ATP and reduce NAD+ to NADH, which then goes through the electron transport chain. We'll see the exact yield of NADH later on in class. It's really important that you memorize that the starting substrate in this pathway is glucose, which is a 6-carbon molecule, whereas the final product of the pathway is pyruvate, 2 pyruvate molecules that are 6 carbons each. Because of this difference, 2 pyruvate molecules are formed. Now let's again summarize exactly what's happening in glycolysis. Remember, the substrate is glucose. That's what enters the first reaction. This is a 9-step process where we have 3 main stages, which we'll talk about in the forthcoming slides. Each step uses a different enzyme and has a different reactant and product. The product of one step becomes the reactant for the next. So remember the difference between a metabolic process and a metabolic pathway. A process can be reversible. So the splitting of glucose, we can split glucose or we can build glucose or transform to glucose again through gluconeogenesis. So the metabolic process is the splitting of glucose. The metabolic pathway is the 9-step process which converts glucose to pyruvate. And recall, depending on the directional arrows, we see that there's reversible versus irreversible reactions. And the irreversible reactions happens at step 1, step 3, and step 9. to ensure we have metabolic control and that the reaction goes to completion. So let's look at what is really happening. So traditionally, glycolysis is represented by one linear line of 9 reactions, which I showed you on the previous slide. But in reality, it's not this simple. Remember that glucose is a 6-carbon molecule, while the product is a 3-carbon molecule. This means that you would expect 2 3-carbon molecules to be formed from one glucose molecule. When the glucose splits in step 4, ultimately we get 2 glycerolaldehyde 3-phosphate molecules, which are formed, and these yield 2 pyruvate. So sources do show glycolysis this way, but most don't. And as a result, students often think that the 3-carbon DHAP, dihydroxyacetone phosphate, formed just gets lost. But instead, it is also converted to glycerolaldehyde 3-phosphate. And now we have 2 pathways that are going on simultaneously to produce 1 pyruvate each. So this diagram is more reflective of what is actually happening. As I mentioned 2 slides ago, there are 3 stages of glycolysis. The first step is a regulation step that starts the pathway, and it's not included in any of the stages. So the first stage is called the priming stage, which encompasses reaction 2 and reaction 3, where we're priming glucose to be split. Then we go to the second stage, which is the splitting stage, which incorporates reaction 4 and reaction 5. Then we have the third stage, which is all about producing oxidizing and reducing agents and ATP. The oxidizing and reducing agents will go to the mitochondria and will be used in the electron transport chain to yield ATP. So the oxidizing and reducing stage is steps 6 to 10, or 6 to 9 inclusive. So now let's talk about the net yield of ATP, or energy. As was mentioned before, step 1 and step 3 consume an ATP. But we generate ATP in 2 steps. This is step 6 and step 9. The important thing to remember is that once we split into the 3-carbon glycerolaldehyde phosphate, everything is happening twice. So the ATP generated in step 6 and step 9 happens twice, for a total of 4 ATP generated per cycle of glycolysis. Then when you subtract the 2 ATP used from the 4 ATP produced, this yields a net of 2 ATP produced per glucose molecule. So if we were to eat an apple and consume 3,000 molecules of glucose, that one apple can yield a lot of ATP through glycolysis. Always come back to the biochemical equation for glycolysis, which is down at the bottom of the slide in orange, where we have 1 glucose molecule plus 2 ATP consumed plus 2 NAD plus consumed leads to the production of 2 pyruvate, 4 ATP, and 2 NADH. So remember that glycogen can also enter glycolysis. So glycogen technically can't, but the breakdown of glycogen into glucose can. And so what happens is that we actually skip a step, that glycogen is broken down into glucose 6-phosphate and enters glycolysis at the second step. So this means that we have 1 less consumed ATP. So this is more efficient energy source. So the net yield from glycolysis from 1 glycogen molecule of glucose is 3 ATP, because we enter the glucose from glycogen enters at the second step, which is glucose 6-phosphate. So therefore, we only have 1 ATP being consumed from glycolysis at step 3. Then we have the production is the same in step 6 and step 9. So we have 1 consumed plus 4 produced is equal to 3 net ATP. So glucose coming from the stored form of carbohydrate, which is glycogen, has a higher energy yield. It can produce 3 ATP and not 2. This is a bit of a redundant slide from the previous one, but it showcases the entry of glycogen into glycolysis. That remember, glycogen is the animal starch. It has repeating monomers of glucose, and that's the stored form of carbohydrate. So when we have an excess amount of glucose within the cell, we store it for when we need it in a very energy-dense format known as glycogen. So glycogen, when it gets broken down into glucose 6-phosphate, and that's where it enters glycolysis. So it skips the first step of that energy-consuming step where glucose gets converted to glucose 6-phosphate. So the equation for glycolysis from glycogen is up at the top of this slide. So we have glucose 6-phosphate is the substrate, plus 1 ATP, plus 2 NAD+, yields 2 pyruvate, plus 4 ATP, plus 2 NADH. So we reduce the number of ATPs that are consumed, and therefore the net yield of ATP is 3, not 2. I would like you to pause the recording right now and complete this slide on your own. So I want you to go back through your notes and summarize the inputs and outputs of glycolysis by looking at the 9 chemical reactions that we went over on the previous slide. So remember that the chemical equation of glycolysis is glucose plus 2 ATP plus 2 NAD plus leads to 2 pyruvate plus 4 ATP plus 2 NADH. And that's the process. That's what's happening. That's the input and the outputs. But through the pathway, we have a boatload of enzymes. We have a whole bunch of different reactants and products, etc. So let's look at the process and the inputs and outputs of the process. The input is the substrate, specifically is glucose. Then we have 2 ATP being consumed, which produces 4 ADP. We have 2 coenzymes known as NAD+ that are being consumed. And the outputs are 2 pyruvate molecules, 4 ATP, 2 ADP, 2 NADH, and 2 water molecules. Don't forget about those water molecules. You can see them when you look at the 9 chemical reactions that we have on the last slide when we looked at the metabolic pathway. You need to know each step, the substrate, the product, the reaction type, the enzyme name, and the summary of the inputs and outputs of the pathway, which we will go over more this week in the synchronous and asynchronous content. Now let's talk about the regulation of glycolysis. How fast is glycolysis and which reactions are the slowest? Well, one of the slowest reactions within glycolysis is the reaction involving phosphofructokinase, or PFK, which is the third step. And this is called the rate-limiting enzyme glycolysis because it's the slowest enzyme in the chain of the metabolic pathway. It is allosterically regulated. That means it receives feedback about the energy status of the cell, the AMP-ATP ratio, the ADP and inorganic phosphate concentrations, as well as something called citrate concentrations. And these either accelerate this enzyme and catalyze it, or they inhibit it. So remember, we talked about competitive and non-competitive inhibition. This enzyme is sensitive to fluctuations in the energy status of the cell, and therefore the rate at which the reactions happen will be affected by the energy status. It either accelerates PFK or decelerates and inhibits PFK from happening. With exercise training that utilizes glycolysis, what we see is that enzyme activity changes, specifically the rate-limiting enzymes. And in this case, it's phosphofructokinase. We see that its affinity for the substrate improves. That means it doesn't require as much substrate to get to half its maximal velocity of the reaction. Therefore, we can shuttle through glycolysis faster. It kind of comes down to that saying, whereas fast is our slowest link. The same goes with glycolysis. Phosphofructokinase is the enzyme that's the slowest within this metabolic pathway, and therefore glycolysis is as fast as that enzyme is. So with exercise training, we can increase the affinity of PFK for its substrate and lower the Km. As well, we can increase the number of this enzyme. Therefore, we're not limited as much by enzyme saturation. Let's talk about PFK, or phosphofructokinase, a little bit more. Specifically, the isoform is number 1. This is considered the rate-limiting enzyme of glycolysis because it is the slowest step. It's the slowest enzyme that has the highest Km. That means it needs a lot of substrate in order for it to get to half its maximal velocity. It is a big protein. It has 4 subunits. So because it has subunits, we know that it's in a quaternary state. It's a big protein. It also is allosterically inhibited. And this means that the regulation of this enzyme is by binding an effector molecule at a site other than the enzyme's active site. So this is very much similar to a non-competitive inhibitor, where we see binding to an area outside the substrate binding sites, which it inhibits or accelerates the enzyme. So this is inhibited by high ATP and low pH, and it's activated with high AMP concentrations. That means when the energy status of the cell is very low, it activates this enzyme. So the cell can increase or decrease the rate of glycolysis in response to the cell's energy requirements. In summary, where is glycolysis found? It's in the cytoplasm or the cytosol. Why do we need it? Well, it's the first step of glucose oxidation. It's the first step of breaking down glucose to yield energy. It only has 9 enzymatic steps, and therefore it's very fast. So it creates ATP from glucose. What is involved? Well, it's a process that splits glucose into 2 pyruvate, 2 ATP, and 2 NADH, that the NADH travel to the mitochondria to yield more ATP in the electron transport chain. Glucose oxidation happens with blood glucose or glycogen. It depends if we're fed versus fasted or rested or exercising. When does glycolysis happen? Well, it happens constantly, all the time. The rate or the speed of glycolysis depends on the demand for ATP, and this is where you question rest versus exercise. It has 9 enzymatic reactions. Right here it says 10 because you could count the transformation, the isomerase reaction that converts it as a step, but we don't count that. So you can correct that in your slides that there's 9 enzymatic reactions. So it's relatively fast metabolic pathway. As well, the energy status of the cell, the AMP/ATP ratio will determine the flux through the pathway. So ATP we can break down 2 times. We can break it down to ADP, where we have diphosphate, and then we can break the ADP down to AMP, monophosphate, where there's only 1 phosphate molecule. So when AMP is high, that means we've been breaking down ATP a lot. And so the energy status of the cell is very low. And this acts to activate phosphofructokinase, the slowest step in the metabolic pathway, to get glycolysis going. This concludes this mini lecture.